these for so long. We know what works best for certain organisms. You can have complex media, which contains materials that vary in chemical composition. That would be things like blood or yeast extract or beef broth. So these are things that, even though you know what's in it, there's still some variation. I mean, yeast extract. When we're feeding yeast, you give them yeast to eat. And there is no saying that the yeast are going to be exactly the same every single time. But they're close enough that you just can go with it. But that makes it a complex medium. Sometimes you need the blood, the beef, the yeast, because that will support a wider range of microorganisms. This has actually become an entire industry, having the fetal bovine serum or whatever, so that for feeding the mammalian cell cultures, because they're so picky about any slight little thing, and they'll die. So media is incredibly important. Different medias will mess everything up. Yeah.
What exactly is a media?
A media is just a nutrient broth, basically. So it's anything that's going to provide nutrients to grow the microorganism. Since I work in yeast most, that's the one I can tell you a bunch of. So you have like a rich media, which has everything and anything will grow on that. And then we'll talk about like selective medias, where you can take certain things out of it. So it's just whatever will make your organism grow. Basically, usually you need some kind of energy source and a carbon source. Okay. Selective media encourages the growth of some organisms, but suppresses the growth of others. So there are multiple kinds of selective medias, E. coli-wise. We can have rich media plates, which all E. coli will grow on, and then we can have, say, E. coli plates that have an ampicillin added to it. So we've added an antibiotic. And so only E. coli that has an antibiotic resistance gene can grow on those plates. The selective media. Only some colonies can grow. So that's incredibly useful in science. A lot of times when we're giving them plasmids, we add something so we can select for them. Differential media produces observable changes that make it easier to distinguish the desired organism from others. So this is something when you guys have to figure out your microbes later, you'll be doing and playing with. Just certain medias, certain organisms grow better on certain medias. And so if you can start to figure out, have some kind of observable change, it'll help you figure out what organism you're looking at. Enrichment media. So some organisms do not like to be cultured and grown. There are a lot of microorganisms we haven't been able to culture. If you think about all of microorganisms living in the deep sea ocean, they're used to high pressure. If we bring them up to sea level, they can't survive. And so being able to culture them or learning how to culture them is challenging. But other ones that need some kind of special nutrient, they need something that will turn on a gene, turn off a gene, something. You can do enrichment media, which will just really encourage the growth of maybe some of those trickier microorganisms. So some examples. Selective media can also be pH. You change the pH, you change what's going to be able to grow on it. So if you make it a nice, happy, neutral pH, 7.3-ish, everything can grow. You're going to get tons of bacteria growing on that. You start dropping that pH, the bacteria aren't going to survive as well, so you're only going to start getting fungal colonies. You'll have some fungal colonies on the regular pH, but bacteria replicates so much quicker, they will outcompete the fungus. Okay, so example of selective media. Okay. A differential media. So here we have, you do streaks of two different things, you're trying to see a difference. And here is a great difference between E. coli and salmonella. And so it's one of those things, by putting them, streaking them onto this plate, you can very clearly see the difference, which you can't see in the nutrient auger. So this one will just let everything grow. But in this one, you get very distinctive colors and differences which help identify the organism. Or you can even have one which doesn't allow it to grow, which is another way of identifying. So kind of as a selective agent. Does this make sense? Visualize some of the things we're talking about? Growth media. So, some of the organisms are anaerobic. And so if you're trying to grow anaerobic organisms, it makes things tricky because you need special conditions. You need to remove free oxygen from the media, and you need to be able to actually incubate the plates in an environment without oxygen, which ends up being a little hard. And so here's just kind of a little example of one of the containers that they have to actually help you remove, it's catalyzing reactions, so that way you can remove the oxygen. You have indicators to make sure you don't have oxygen, so you know your plates are fine in this tube. And then you actually just pick the entire container up and put it in whatever incubator you need to give them the temperature. Make sense? Question? Microbial growth. Okay, we've talked about growth, we've talked about doubling. There are four basic stages. You have lag, log, stationary, and then eventually death. Okay, so these are very important when you're trying to deal with cultures and when you want to look at them. So generation time is just the time required for a bacterial cell to grow and divide. And that generation time changes from microbe to microbe. And so this is just kind of to give you an idea. So we have, with every one of them, we always look at it in log. So you have this lag time. So if you put a single colony into liquid media, it's going to have this lag phase where it kind of doubles, not really doing much. You won't really see any change. And then all of a sudden, it's going to shoot up. It's a logarithmic. Okay, it's exponential phase. And it is going to double like crazy. Most experiments you want to be doing at this stage. When we were looking at the endospores, we were looking at this stage because we were wanting them to be stationary. We wanted them to use up all of their nutrients and start being in a spot where times weren't so good, so they wanted to make those endospores. So, most cultures we look at, they're in log. They're happy, they're dividing, they're good cells. That's why they're easy to stain and look at. But some of them you need that stationary, media starts running out, or they get too old, there's no food, you start getting into your death phase. Okay, so going through those again real quick. Lag, no significant increase in numbers. They're active, but they're kind of getting used to their new environment. You've dropped a toothpick full of cells into 5 mils of rock. They don't really know what to do with that. Log phase. They've gotten over their shock. They're growing like crazy. And this is when they really don't take long. Coli doubles every 20 minutes. If it's happy, yeast, if it's happy, can double every 2.5 hours. Human cells aren't really that happy, so they usually take about 24. But just kind of give you some idea of how long it takes to get a complete doubling. So, I mean, every 20 minutes you're getting doubling of E. coli. It makes sense why overnight you have so many cultures. Okay. Stationary. They keep producing but the old cells are starting to die. All cells have a replication limit. After they've hit so many cell divisions, they kind of stop dividing and they die. They're considered old. So different cell types have different cell numbers before they decide that that's it. Nutrients is running out. Also, waste starts to really accumulate. So it's one of those issues. You don't have food, you have too much garbage, not really a good place to grow. And then death phase, you aren't dividing anymore, you're only dying. So how do we measure all of this? There are multiple ways of measuring cell growth. You can spread them onto a plate and count colonies. You can do filtration, where you can concentrate the bacteria on a filter and then grow those. Or we can also study the turbidity. So how cloudy is that liquid? How much light can we pass through that spectrophotometer? Or you can do an actual direct microscopic count. So what do these look like? Plate counting method. It is one of the most common. It is pretty easy. It's kind of time consuming. But the idea is you do serial dilutions until you get to a reasonable number of colonies to count. And then you know how many colonies you have here. You know what dilution factor that was, so then you know how many colonies you have in your original by doing some back math. Okay, so you basically just keep doing dilutions until you get a reasonable number of colonies, and then you count backwards. A lot of times you do get those too many to count. You just kind of look at it and go, yeah, we're not touching that. And if you get, and even though 6 sounds like a nice number to count, it's really not enough to be very accurate. So you're going to want to be in the 65 to anywhere between 65 and under like 300 is a good range. It's easy to count. You feel confident in your numbers, and then you can scale back and figure out what was the actual CFU, colony forming units, in that original culture. So the viability a lot of times is measured in CFU, just how many of those cells were viable and capable of making microorganisms. So, membrane filtration. Okay. You take a sample, you filter it through a membrane. So this can be good because you can do different levels of membranes if you want, but for the most part you're probably going to catch everything that goes through because filters are going to be smaller than your microbes. So you can get nice clean broth out the bottom, take that membrane, put it onto rich media, and colonies are going to come up. And so you can actually see little tiny spots of colonies. It's a nice little picture of what colonies look like with a filter underneath it. And then, so if you need to actually know the number of colony forming units, most often you're going to do the plate just for any assays. But if you just need to know approximate where you're at, make sure you're in log phase so you can do experiments or you can know where growth is, we're going to do spectrophotometry. Just because this is as easy as taking 2 cultures, or 2 samples, a blank, which is your media with nothing in it, and a sample of your media with your cells in it. And you read how much light can pass through, and that tells you how many cells you have in that culture. So, you're blank, you send it through, light goes through, it senses the absorbance, you get a number, that's your blank, that's your new 0. You put in your culture tube, your inoculated broth culture, and all of those little cells are going to start scattering the light, so not as much is going to get to the sensor, so you can start seeing what the absorbance is, and based on those absorbance, you can tell how many cells you have. You use different wavelengths of light based on what cell types you're using or what you're doing. Most of the time, cell cultures are at a wavelength of 600, just because that's what they have found to be good. And so it's one of the most easy, common pieces of scientific equipment. Okay? Is this making sense? Okay. Cell counters. If you're doing something you need to be very, very precise about, you need to know how many cells you have in your culture. So for you guys, if you're doing something medically and you need to know how many cells, how many viable cells, a lot of times you might actually end up having to do a cell counter, and this is where you look at them through a flow cytometer on an actual microscope. And so you actually count how many cells you see in a region. So you can have it, you can see, you can actually start counting them. Gives you nice grid lines so you can see it easier. That is a possibility. It's a lot more frustrating, but it is something that is done, and it depends on the circumstances whether or not you would actually have to do that. Okay. So, we've cultured microorganisms, we've counted microorganisms, then storing microorganisms. Most common way of preserving microorganisms is throw them at 4 degrees, put them in the fridge. It slows them down. They can keep for quite a while depending on the organism, but basically usually the agar dries out before the organisms are really too bad, so you end up chucking them because they've dried out. But you can keep, they're good for several weeks this way. If you really want to keep them for years and years and years, you start going for that deep freeze. So this is usually you add glycerol to make it so they don't actually freeze. You don't want the organisms to freeze. Why wouldn't you want it to freeze? The water, because if the water actually freezes, then it can become, ice is kind of sharp. So, I mean, it is a method of breaking open E. coli, is just to freeze and thaw it a bunch of times, and it will burst open all the cells. So if you're actually wanting to retrieve this microorganism later, you want to usually put it in glycerol so that it won't actually freeze, but it's at such cold temperatures that it's not growing, and it is basically in stasis. So in lab, you keep it at like negative 80 degrees. So you have like a negative 20 for the couple year storage, negative 80 for the long-term deep storage, or even liquid nitrogen at negative 70. So all biotech companies keep their stocks at that negative 80 degree temperature, just serious deep freeze. Another option for some microbes, you can do lyophilization. So lyophilization is you put it in basically a fancy centrifuge that has a vacuum, and it's going to pull all of the water away, and you end up getting kind of a freeze-dried culture. I haven't played with many organisms that can actually survive this. Usually this is more done for DNA. Like if you order primers and stuff, the DNA, it can be very safely shipped, lyophilized, and you don't have to worry about it. But you can lyophilize some microorganisms. Okay. So, basic storage, 4 degrees short term, freeze it if you need it long term. Some of them can be lyophilized. I mean, if you think about it, we've had some things that have been taken out of tombs in Egypt and have been grown again. They basically got lyophilized. They got all of the water removed from them, not in the process we would do today, but and they were able to be brought back. So some organisms clearly are capable of being rejuvenated after more than decades. Okay, and I want to show you this. So this is a slant which we will be playing with today. So I just wanted to quickly give you, in case there was anybody who had any questions of what a slant was. But that's chapter 6, so I think we'll pause there and we'll come back. You'll do chapter 5 tomorrow and Monday, and then we'll do 7 on Tuesday, Wednesday, and then you'll have your test Thursday. This class is apparently full of tests. You have 5 tests in 8 weeks. Well, 5 lab tests and 2 lab practicals, so. Are there any basic questions on growing cells, culturing cells, pure cultures of cells? Anything about how to handle cells, because that's what we're going to be doing in the lab.